Good afternoon, everyone. So I'm Andrew McPherson. I'm a PhD student, and I'm currently in SFU doing computing science. And I'm in Dr. Saurabh Shah's lab, co-supervised by him and Cheng Sayanel at SFU. So I'll be giving an update, basically, titled Computational Interpretation of Cancer Genomes. So the guiding principle in our lab is that cancer discovery through sequencing assays necessitate advances in computational methods. I think it's important to understand and reiterate, even though it's been said before, why these problems are fundamentally hard. And I think one of the main problems is just the sheer scale of the sequencing data that we're interested in generating and interpreting. So for instance, say we have 100 patients with a particular type of cancer. We're interested in interrogating those tumors for common properties. It will help us understand the biology. So we generate 100 associated data sets. So one of these data sets, in order to overcome the problems of heterogeneity in the tumor and normal contamination, needs to be at least 500 million short read sequences in size. To give you an idea of the scale of this data, it's, if you were to write it out in 12-point font, it would get you to the moon and back 30 times. So we don't actually do that, but just to give you an idea. On the flip side, what we're actually looking for is perhaps a handful of sequences in that huge data set that contains one particular mutation that is causative of this cancer or is driving the tumor metastasis progression. So with the scale of the sequencing data is increasing, and of course, we know that that scale is outpacing the increases in computer efficiency. And so we can't just throw more computers at it, and we can't wait a few years and be able to throw better computers at it. We have to develop new computational methods now. So we've had some success in doing this recently, and I think one of the reasons we've been reasonably good at this is because the people who are developing the machine learning, statistical models, and data mining techniques are collaborating as closely as possible as the people who are asking questions about cancer genomics, tumor evolution, and genomics of disease. And so we like our bioimplementations to be needy in real-world data, so they're not working on, say, simulations or cell lines. And this has helped us publish some computational advances in somatic mutation detection, for instance, copy number alterations, LOH detection and rearrangements, gene fusion detection, and mutation interpretation methods. And all of these computational advances have led to some interesting discoveries in cancer, such as the FOXL2, ARID1A, and DICER mutations in rare ovarian cancers, EZH2, C2TA mutations in B-cell lymphomas, 1433 fusions, and C2TA fusions in other cancers, and also our studies on mutational landscapes and tumor evolution. So I'll just go into a few details of what this data looks like and what these four different general techniques look like. So for somatic mutation detection, our most recent method is called MutationSeq. It's a feature-based classifier of these single nucleotide mutations. It allows us to discern, importantly, allows us to discern subclonal mutations from artifacts. Subclonal mutations are often characterized by low evidence, but they're the ones that are very interesting from the perspective of studying tumor evolution and in a clinical setting, because these subclonal mutations could be the ones that are selected for during the course of therapy and become recurrence, part of a recurrence. So in the picture here, we have 104 features for 3,000 of our training events, training SMBs, distilled down to 3D space. And we can see with this method, using these features, that we can separate the somatic SMBs that we're interested in from the germline variants and artifacts. Continuing, we have the work we were doing on copy number and loss of heterozygosity, detecting, for instance, amplifications and deletions of genomic regions in the genome. With HMM copy, for instance, we can detect identify regions where, for instance, here, this plot is a plot of allelic ratios of heterozygous SNPs, single nucleotide polymorphisms. Since they're heterozygous, we expect an equal proportion of the reference and non-reference base. But in this case, for this region, we're getting either all reference or all variant. And that's telling us that we've lost one of the alleles for this entire arm of chromosome 20. Since, interestingly, this area is also copy neutral. So that means we've gained one allele, had duplication of one allele, and lost the other allele. Finally, my own work is depicting rearrangements and gene fusions. And so I've developed a package called Diffuse that looks for fusion transcripts predicted in RNA sequencing data. And we've had a number of successes recently in identifying biologically relevant fusion transcripts in some rare cancers. Also recently published is a tool that allows us to understand the underlying genomic rearrangement, possibly complex genomic rearrangement, using a joint analysis of the RNA sequencing and genome sequencing. So in the bottom picture, you see these 4 different genes from 3 different chromosomes are coming together simultaneously. Simultaneously, they're broken and rejoined to create a picture on the bottom right, where we have the creation of these 4 fusion genes. 2 of these fusion genes are expressed as fusion transcripts, and one of them involves this specific conflict overexpression. Finally, we like to take the different variants that we predicted using the previously mentioned tools and understand how they affect tumor biology by understanding their effect on gene expression. So to do this, we've just published this method called DriverNet, which takes genomic aberrations of classified by genes, so genes, aberrated genes in patients, and we combine that with an influence graph, which tells us which genes influence the expression of other genes. We combine that further with the gene expression of genes in our patients, where we can identify with this gene expression matrix particular genes that are significantly overexpressed or underexpressed relative to other patients. And we can postulate this combinatorial problem where we try to identify the genes that are aberrated that most influence the expression of the most other genes. Basically, we're looking for trends, effects of these genomic aberrations in terms of influencing the expression of various genes. This has led to the discovery of a novel driver mutation in DLCL. Finally, I'll talk a little bit about the grant that is funding a large majority of our work called Computational Interpretation of Cancer Genomes, Defining Computational Landscapes for Translational Genomics. So the first aim of this grant is to develop new methods where the current methods are lacking. The second aim is to evaluate the implementations of these methods and the other people's implementations, basically do an unbiased analysis of which is the best, a benchmarking of which is the best method. And obviously this evaluation that we do in aim 2 can help with aim 1, where we try and develop the most robust method in an iterative procedure. Finally, or thirdly, we have this with third aim, we'd like to create cloud compute ready, modular, and distributable workflows to distribute them to the community. And the key motivation behind the third aim here is that a lot of good software or a lot of good techniques are published by a graduate student who works on them very hard and then puts a prototype on the internet, and that definitely doesn't lead to clinical grade software. So here we're taking good algorithmic ideas